All right, so here's part 2 from class on March 26th. In the last video, you saw that we were trying to see what would happen if we crossed or reproduced a heterozygous pea plant for the height gene with a heterozygous plant for the height gene and what we would get. So what we do in order to predict what kind of offspring we're going to get is we're going to use statistics. Now, statistics is like a guess of what's going to happen in the future based on what we already know now. And how we do that is by making a Punnett square. So there was a man who lived whose last name was Punnett, and he invented this way of making a prediction to see what possible offspring could we get from these two parents. So here's how we're going to do this. I'm going to be showing you how we can use the parent genotypes to write them onto the Punnett square so we can predict the offspring. So let's call this plant parent 1. And I'll put them in red. And then let's call this plant parent 2, and we'll put them in blue. Okay, so parent 1, we're going to take a look at their that should be in red. We're going to take a look at their genotype, okay? Genotype of parent 1, and we're going to put their genotype on the top of our Punnett square. So we have a capital T allele. We also have a lowercase t because parent 1 is heterozygous for the height gene. Parent 2, we're also going to do the same thing. We're going to take the genotype of parent 2, but we're going to put it on this up and down side. So we're going to start with our capital T because we get one capital T from this plant, and then we also can get one lowercase allele from this plant. So this is telling us if this plant were to make sex cells, one of its sex cells could have a capital T, and one of its sex cells could have a lowercase t. Because remember from our bio reads, we learned that when this plant is making its gametes, its sex cells, its gametes are only going to get one of its two possible alleles, so either a capital or a lowercase t. Same thing for parent 1. So here's how we do our Punnett square. We're going to start off by going to the right. So we're going to fill in our boxes to try to see what percentage of our offspring from these two parents are going to have each genotype. So we're going to start off on this side, and we're going to carry over our dominant allele from parent 2 all the way to the right. That's a capital T. The same thing for our lowercase t. We're going to carry our lowercase t all the way over to the right and fill it in here. So we filled it in that way. Now we want to fill in for our parent 1. We're going to go down. Okay, so we're going to start with capital T, and we're going to go down, and we're going to go down. Now the reason why I put this red one in front of the lowercase t is because we always write our genotypes with the capital letter first. So here, if we have our lowercase t, we're going to bring it down, and we're going to put it second because remember, we always put our capital T first, and then we're going to bring this one down, and it doesn't matter what order you put these lowercase t's in because either way, you're still going to have a homozygous recessive genotype. Okay, so this is how we make a Punnett square. Pause the video and watch it one more time if you want another explanation. So now that we know what our genotypes are, let's take a look at the percentages or the number of offspring that we get for each type. So remember, what this Punnett square is telling us is that if these two parents had 4 kids, just 4 babies, what are the chances that each baby would have this genotype, and this genotype? So it's possible that these plants could actually have 100 babies or 1,000 babies. But what this is telling us is that if these two plants had 1,000 offspring, only 1 out of 4 of the 1,000 offspring is going to have this genotype. Whereas, look here, we got half of our offspring have the capital T lowercase t genotype, which means if we had 1,000 babies, half of those babies would have these genotypes. Why? Well, that's because for our capital T, we got 1 out of 4 have the capital T genotype, and if we wrote that as a percentage, that would actually be 25%, just like we count quarters, right? One quarter is 25%. So then what percent of our offspring are heterozygous? Well, if you look here, 2 out of our 4 are heterozygous. So convert that into a percentage, that would be 50%. Continue along with the capital T, lowercase t, tall and short. Try to figure out what are the number of offspring for each genotype and each phenotype. And then for the last thing, if you want to do the Kahoot that I posted on here, you can get some really good practice in there and submit your